Let's go.
Hi, everybody. We're back here again. I'm Dr. Carlos Herrera, and I'm joined by Dr. Aliffe. Dr. Aliffe is a researcher, a physician, a great doctor that has been doing a lot of study in the field of cannabis and cannabis treatments. And I am sure that he is much better capable of explaining what he has been doing in the last couple of years than myself. So why don't you tell us, Doctor, a little bit of yourself?
Sure, Dr. Herrera, thank you. And muchísimas gracias a todos por invitarme a esta conferencia. Y me alegra mucho que México está como haciendo los primeros pasos en crear un ambiente en que la gente que aprovechan del cannabis como medicina, como algo para ayudarles con su salud, que México es como Canadá, está como está marchando al futuro, como con mucha como energía, pero también enfoque y el gobierno está, me parece, está a su lado, no como en los Estados Unidos. ¿No? Pues ustedes son los líderes. Now, regarding my background, so
Sure.
my interest in medicine originally, when I did my undergraduate degree at Harvard in medicinal chemistry. And I did cancer research at the time. And the cancer research was actually looking at natural compounds that impact the carcinogenesis process, specifically tumor promoters. And as it turned out, years later, decades later, really, I'm probably older than you think I am, that same system of tumor promotion involves an enzyme called protein kinase C. Protein kinase C sits right next to the cannabinoid receptors.
Okay.
And so I was working in the same space, but it was before the cannabinoid receptors were discovered. But the main thing is I have always had an interest all the way to the molecular level of how medicines work in the body, how the physiology works, what the disease processes are like. So when I went to Stanford for the MD PhD program in cancer biology, I continued that path and I did fellowship in clinical pharmacology, training in anesthesia. And so that has kind of formed my medical perspective on the importance of observing the patient continuously and recognizing the connection between mind and body, which is super evident in anesthesia. So in the cannabis space, it's only natural for me to be able to look at the complexity of cannabis from a molecular level and also the complexity of the individual's response, each person perhaps being different than the last. And so it's a very exciting area to me. It's super important. I think that the 21st century, the future of medicine is really around our expanding understanding of the endocannabinoid system and how cannabis and cannabinoids interact with it.
Great. So, Doctor, look, there's been a lot of talk all morning about the applications of cannabinoids, but very specifically for chronic pain, for neurodegenerative processes, and nobody has dared to touch the subject of cancer.
Okay.
Right? Okay, so
Yeah.
only, yeah, it's, I mean, they have brought it up that it always goes, you know how it goes. They go like, yeah. Well, even though there's no research, actual research that proves that it can improve these certain conditions with cancer, it can help certainly in the treatment amongst the chemotherapies and the radiotherapies for the well-being of the patient, right? But in your research, have you have any findings except more than only the regional places where the cytokines and the endocannabinoid system work?
Yes, well, as I mentioned, my interest typically is from the molecular level and the mechanistic level. And so there are two things that jump out about that. So one is that cancer itself is primarily an immune dysfunction condition, and that appears in the latest literature, at least, to be linked to immune dysfunction. So you have everybody, all of us in the audience, I probably shouldn't say this in the middle of a pandemic, but all of us in the audience, because we're alive, we're producing new cells every day. And because the human body is not mechanically perfect, and because there are, billions, trillions of cells in the body that are renewing themselves, every so often there are errors that are made in the production of those cells at the genetic level, at the level of genetic expression. And so some of those can end up being what are called transformed cells that can proceed on to become a cancer. As well, viruses and other foreign genetic material can affect human cells in such a way that they become transformed into a malignant cell type. But once that transformation occurs, the statistics, the figures that come out of the literature primarily are, say, for instance, in the case of breast cancer, that it takes 15 to 20 years for that initial cell, that's called the transformed cell, to progress to a state where, as physicians, we can detect and diagnose it as a cancer. That's a very long time. So throughout that entire time, that cell, and as it's growing into more and more cells in a large enough mass to be detected either on physical exam or radiography, ultrasound, that group of cells has to completely evade the immune system. The cancer is really supposed to be cured by our immune system, because that's the reason why most of us never get it. Because normally what happens to those individual cells is that they don't survive 15 or 20 years, but they may survive 15 or 20 minutes or 15 or 20 days, but they don't survive long enough to have a physiologic impact on the body. And natural killer cells and other cells from the immune system, that is their specific task, to eliminate those cells before they grow into an actual palpable mass or a disease that we call cancer. So carcinogenesis is the issue. So on the one hand, cannabinoid compounds, because of their activity, not only at the CB1 and CB2 receptor, but at multiple other receptor sites throughout the body, like the TRPV1 receptor, have an immune modulating effect. And so they can both enhance the ability of the immune system to eliminate cancer cells, as well direct action on cancer cells by cannabinoids can promote what's called apoptosis, or self-destruction, cellular self-destruction of those cancer cells. So that is, people would call that preventive, and that is a little bit different than a person who already has cancer, and then the aspect with respect to curative effects. But there is also literature regarding the potential curative benefits of cannabis-derived products. And when I say cannabis-derived, I mean in the sense of extracts as opposed to individual cannabinoids. There's a study right here that I have on my laptop that says, the title of it is Appraising the Entourage Effect, the anti-tumor action of a pure cannabinoid versus a botanical drug preparation in preclinical models of cancer. And what they found actually was that the entourage effect, which is the combination of multiple bioactive compounds that are found within extracts of cannabis plant material, typically CBD, THC, other cannabinoids, but also terpenes, for instance, beta-caryophyllene, which you probably heard about earlier from Dr. Philip Blair. So even just with these three compounds, you already have an entourage effect. So for people that are wondering, is the entourage effect real? Yes, it is, because you have synergy, you have complementarity, you have activity at multiple sites throughout the body. But what they found in this study was that the botanical drug product actually was more effective in initiating regression of tumor mass, so the killing of tumor cells, and it was more effective than just the isolated cannabinoids.
Could you walk us through a little bit the interaction, the mechanisms in which this works, the entourage effect? For instance, does an entourage effect improve the acceptance of the TRPV1 receptor, or does it make the connection from the cannabinoids endure a little bit more?
So yeah, so there's and the reason I mentioned the immune dysfunction issue is because the endocannabinoid system regulates immune function. And most of us, modern humans living in, especially say a place like Distrito Federal, in a big city, Los Angeles, Bogota, Miami, places like that. But just modern life in general, adult humans especially are subjected to chronic stress. The lifestyle, the culture, also our lack of exposure. Yes, air pollution. So there are carcinogens in the environment. There are carcinogens in the general como ambiente that we're exposed to, but also there are compounds that affect or irritate the immune system and other aspects of human physiology. So this can cause a dysregulation. So chronic inflammation, I often try to explain it. You mentioned we have a diverse audience. Some are physicians and people with different disparate backgrounds. That chronic inflammation in the human body is like a lot of little fires, like we see often, say in California, that there are these wildfires in different parts of the state, and they're going on and they're destroying local areas, destroying individual houses, or destroying individual areas that are forest land. But the entire body of California overall is still continuing. It's not dying, but it is definitely affected by these small fires. Well, inflammation basically means fire inside of the body, a fiery process. And so anti-inflammatory agents, for instance, cannabidiol, for instance, at the CB2 receptor activity, beta-caryophyllene, and also within the HPA axis, dopamine, through CB1 activity, THC activity at CB1 and CB2, TRPV1. So there are a bunch of modulators. I mean, these are just names, but basically to understand the function, that the job of the endocannabinoid system is basically to help us get over stress. And as much as people don't like to refer to the recreational usage of cannabis as marijuana, the fact that people use that to reduce their stress levels, and maybe they take a higher dose that then pushes them into a space where they are quote unquote high or intoxicated. But the same thing happens with anesthetic drugs, right? You can make somebody sedated, but they're still awake. You can put them to sleep, but they're breathing spontaneously, or you can put them out completely, so they're unconscious and not even breathing. And of course, you can, put them into a coma. So these are just dosages, but you recognize the clinical pharmacology along the way, that drug or a combination of molecules or agents are acting in a certain space. So there is a concert of function among the various compounds. I mentioned cannabinoids, terpenes. There's also another class of flavonoids that are called canflavins that have been shown to have greater anti-inflammatory activity than non-steroidal anti-inflammatory drugs. So this is definitely an entourage, and it definitely addresses this issue of inflammation, chronic inflammation, running unchecked in the body and helping the cannabinoid system to dial down that inflammation. Because remember, that inflammation, it's not like, maybe it's a bad example. I'm a person who thinks in analogies, but if somebody eats like, habanero chile, it's very hot and it can burn the mouth. It can feel burning in the throat or in the stomach, especially somebody who's not accustomed to it, maybe tourists or whatever. But that's because there's a direct irritant effect of that substance. Yeah. Interestingly, which also interacts with the endocannabinoid system and the opiate system. But so it actually has some health effects and analgesic effects. But the point is, so people are used to thinking of a substance from the outside coming in and doing harm to the body. And so that feels like fire, right? So it feels like there's fire inside of the body from a very hot chile. But this fire
Yeah.
that's in the body is being caused by the cells, right? The cells are releasing what are called pro-inflammatory molecules like cytokines, and those are actually doing damage. So it's really an autoimmune process, cancer is.
Yeah.
It just doesn't get called that because it's got its own institute and it doesn't want to get lumped in with lupus and all these other diseases that are autoimmune. And what one finds in a person when you look systemically is that in addition to the cancer, that the majority of people with cancer also have depression, and that both cancer and depression are associated with inflammation. So neuroinflammation in the case of depression, but also in the case of neurodegenerative diseases. So we have these little inflammatory processes occurring in different parts of the body that, if left unchecked, can manifest as cancer, can manifest as mood disorders, can manifest as neurodegenerative diseases. But the work hasn't been done in that space to where, you know, the global physician community feels confident and comfortable making a concrete statement saying that this is what it's about. But the science is pretty clear.
Great, thanks a lot. And
Sure.
for a follow-up, could you give us like a little bit of a walkthrough of what has been your role in medical marijuana product research and development, and seeing what are your targets, what is your main objective if you're looking for searching for different compounds, or using the entourage effect for certain specific conditions, or a little bit along those lines?
Sure. Yeah. So there are a couple of things that may come across as a bit unique. So as I mentioned, I look at medicine from this standpoint of molecular physiology and pharmacology to systems physiology, then to the pathophysiology, and then to disease manifestations. I prefer to start from the bottom, from the quantum level, if you will, and work up as opposed from the top down. The reason for that is because basically a diagnosis of a disease, so that category that is a disease, and the process we go through the diagnosis is based on several assumptions. Okay, so we look at different studies, we look at signs and symptoms in a patient, lab studies and radiographic studies to determine that a person has this condition. Say, for instance, in the case of diabetes, we may look at hemoglobin A1C and blood sugar, and maybe even do a fasting glucose, those sorts of things, in addition to some other tests. And then we put that person into the category of a diabetic. And then we have certain things we expect from that person that we predict based on our understanding of the progression of diabetes as a disease. And so then somebody might ask, well, and actually there's some very good answers to that. Again, Dr. Blair works with cannabis and diabetes extensively. What I like to look at is, well, what can we do with the endocannabinoid system aside from categorizing this person as having this disease? What can we look at in order to optimize endocannabinoid system function such that the molecular level and cellular level physiologic processes and the pathophysiology no longer produce the signs and symptoms that we associate with the disease? So I'm not, aiming at diabetes by lowering blood sugar, but by, for instance, if there's an inflammatory process involved, like say juvenile diabetes, if there are lifestyle issues, if there are other, if I can find the source of the pathophysiology and see where does that intersect with the endocannabinoid system. Another aspect that's very important that is not often mentioned. So cannabis obviously is a plant. That makes it a botanical medicine. Okay, botanical medicine research looking into the medicinal application of plants. The standard for that approach is to start with characterized plants and to go through a specific extraction process. Usually in the basic early research, there'll be an aqueous fraction to grab the hydrophilic compounds, and there'll be, say, an organic solvent like hexane to grab the hydrophobic compounds, and sometimes a third, for instance, usually ethanol, so then to look at these different fractions and then subfractions and then do bioassays in animals or cells to see what is that activity, what is the activity that's present in this plant. Historically, cannabis has not gone through that process, primarily because it has been seen more recently, say since the 1960s, when Dr. Mashum did his research, as a drug of abuse, as a party drug, not as a medicinal plant. So most of the research, especially in the 60s and 70s, Dr. Mashum had to work with contraband drug material. He was working with hashish. He wasn't working with fresh plants. That means he didn't really know what he was working with or where it came from. But a lot of the basis of our concept of cannabis and in the endocannabinoid system comes from work with confiscated drug material. So that means we don't really know how that material was processed, how it was stored, how old it was. So, imagine with anything else, even if it was a pharmaceutical, and you just found a big bag of pills, in a cloth bag somewhere, and they said, "Oh yeah, go ahead."
No, yeah, I can imagine like trying to trace that particular bar of hashish back to where it came from, and then trying to sort out, like, was it the same crop? Was it a bunch of different crops?
Exactly.
Yeah.
Exactly. So, he wasn't even supposed to. He was kind of going around the law, even though he was getting the hashish from, the police there in Israel. But it wasn't really an acceptable thing to do back in the 1960s. And so, imagine if somebody brings you a bag of pills, 2 kilos of pills in a cloth bag, and say, "Oh, we found this in a stolen car. What is this?" And you give it to a few mice, and they start running around crazy. And they said, "Oh, it's probably like speed or something like that." And it's like, "Yeah, that's what it said on the bag. It said speed." It's like, okay, well, we can do research on speed. Well, you don't really know, what it is. And of course, pills are not that complex. It's mostly chalk and then 1 or 2 molecules in there. But with a plant, so anyway, I mentioned this because of a couple of things. One, they weren't working with fresh plant material. And 2, basically there was an unknown. And 3, because they were looking at it as a drug of abuse, when they were looking for what is the active principle of cannabis, they were thinking in terms of it getting people high. So that's why THC is seen as the cannabinoid and CBD is secondary, even though CBD was identified first. But because it didn't cause intoxication, it wasn't interesting to the people funding those grants and doing that research. So all of that just to say that it hasn't really been approached from a level perspective of what does this plant do in the human body, even in disease models like, epilepsy or diabetes. There have been some clinical studies, but I'm saying from the botanical medicine level where you decide, okay, our in vitro studies show this activity. We think this may apply to epilepsy. We have a rat epilepsy model. Let's, give this to the rats that are from this model. Oh, okay, it looks this way. It looks like it has this effect at this dosage. We can move to a larger animal and maybe move to humans. And of course, because humans are already consuming it, then you have some advanced information. So that's the usual process. And it hasn't undergone that process. So consider when we say CB1 and CB2 receptors, why is that the number 1 and number 2 receptor from the endocannabinoid system? Because that's where they found activity of THC. Everybody was following the THC trail and leaving CBD off to the side, not to mention the other cannabinoids, not to mention the terpenes like beta-caryophyllene, not to mention the flavonoids. So it's not until the 80s that you start to see people identifying the canflavins, Dr. Barrett in London and her group that identified the canflavins, which are flavonoid molecules that, like I said, show more powerful anti-inflammatory effect than non-steroidals like the standard aspirin. So the part of my focus in my own work and to try and influence colleagues is to refocus on the plant and its impact on the endocannabinoid system. Yeah. And part of that refocusing on the plant is to also look at the fresh plant, because fresh cannabis does not have any THC or CBD. THC and CBD are not produced by cannabis, not produced by hemp, not produced by cáñamo. They're produced by human processing of those plants. The
Exactly.
cosecha and the storage of those plants produces that. So when people are concerned about THC exposure, for example, all they would have to do is extract fresh plant material at a low temperature. There would be no decarboxylation. And so you'd have
Exactly.
CBD acid and THC acid. So these are areas that I'm most interested in.
Great. So I guess this forces a little bit this next question. One is being done in terms of making certain that the crops that people are growing for research are specifically the same strain, and amongst the same strain, sometimes the plants itself, they themselves, they have different um, percentages of THC acid or CBD acid. How can you standardize a test of this type of crops?
Well, so this thing about standardization is a human fantasy. There's a huge difference between a living medicine like cannabis and a dead one that comes from the lab like Viagra. So you can control that because you have control in a standard pharmaceutical. You have a specific purified compound and other specific pure compounds that you're combining to make a pill or whatever dosage form. As soon as you have a living material, then you have a dynamic space. You don't have a point on a graph, you have a cloud. And so for people that know, for instance, wine, like red wine production, and even say tequila, that I'm less knowledgeable about than I am wine, depending on the region where it's grown and the time of year that it's harvested and the conditions, it's not just the strain of the plant. It's not just, what substrain of blue agave is it? It's not just, is it Cabernet Sauvignon or Merlot? It is also where it grows and how it's being produced and how it's being processed. And so the same grapes can produce very, very, very different wines across, Spain and Italy, California, whatever. Why is that? Because a plant is a dynamic living organism that is reacting to its conditions. It's not a plastic, Barbie doll that no matter where you put it, if you put it in Alaska, if you put it in the Sahara Desert, if you put it at the bottom of the ocean, it's still Barbie. No, this is a living entity. And that's part of the benefit of cannabis. It's part of the challenge. It means we have to expand our minds to a greater level of biodiversity, but it's part of the opportunity because I know we don't have time to go into it here, but if you look into the botanical medicine aspect, the botany of cannabis and of any medicinal plant. So typically in medicinal plants, the medicinal compounds are what are called secondary metabolites. They're not the basic building blocks of the plants and the carbohydrates, proteins, and things that we may see as having a nutritional value. They're these additional compounds that are produced, and they are produced depending on the condition that the plant finds itself in. And there is a coevolution, some people say, or synergy, collinearity between the plant kingdom, and especially plants that produce, for instance, say, anti-inflammatory compounds like the natural source of aspirin, which can come from white willow bark and other plants, salicylic acid, but also then the diversity of bioactive compounds in cannabis. Those are secondary metabolites that are reacting to the conditions in which it's grown. So growing indoors, hydroponics, outdoors, desert, northern climate, temperate, tropical climate, those are all going to produce different plants regardless of the strain. And, it's, if we just look at ourselves, the human population is just one genus. Okay, we're not really Mexicans and Americans and Canadians. We're just Homo sapiens sapiens, and we're giving ourselves a lot of credit because sapien means wise, and we might just be Homo, and the sapiens is still questionable. but the point is, even if we try to break it down into a subset and say, yeah, well, so it's all the same genus, but these people are Scandinavian and these other people. So take a look at the Maya. So are the Maya just Mexicans? Or are they just Maya? Well, are the Maya in South Central Mexico the same as the ones in Guatemala and elsewhere in Central America? When you do genetic studies, maybe 99% similar, but then all of us are. But you want to look at what is their expression? What is their cultural expression? What are they doing in their daily lives? That might seem like, oh, well, that's another issue. No, it's not, because what the plant is doing in its daily lives, if you give the same strain of cannabis to different groups of Maya people or different groups of Scandinavians, and they take it into their environment and grow it, that plant is going to become Scandinavian indica or Scandinavian sativa. It's going to become Mayan indica, but not just, the same in each of those regions, even though somebody from North America, the US and Canada, part of North America, might say, oh, well, yeah, Mexico, Guatemala, Honduras, yeah, whatever. No, it's not. Those are different regions. The coffee that comes out of those places is different. So why wouldn't the cannabis be different? So we can look at the cultivars and those genetics, and that's important, but we need to look at the expression. So you can take plant samples all along the growth cycle and see what's going on in those trichomes where the primary active compounds are found, and then do chemical fingerprints along the way and at harvest, and of course at the end of processing. The drying and curing, remember, that's about maximizing the decarboxylated forms, primarily THC. THCA is active, and in many ways more active than THC, but without being intoxicating. CBD acid has also been shown in many studies to be more active than CBD. So there's a lot of work that can be done just in that space of understanding, what the plant does, depending on the circumstances that it's in. Because if you take the same species of dog and you raise it from a puppy with a bunch of kids, or you raise it with a bunch of military men that are trained in commando activity, they could be twin dogs, and you put them in those two different environments. Those are very different dogs. So that's the opportunity, but it's also the challenge.
Yeah, I understand perfectly what you're saying. Actually, that's exactly what I'm wondering. Like, how must one would go about doing research with this huge variety? You mentioned we have to open our minds a little bit, right?
Yeah, just have to be a little bit more perceptive, because say an example of those two dogs, which one of those dogs, if you're going to give your son a dog for, his birthday or something, do you want to give him the military trained dog, or do you want to give him the dog that grew up with children and playing in the park? They're not going to have the same expression. But if you really wanted to test, you could test in advance and take those dogs and put them through some tests and see how aggressive are they, how friendly are they, how well trained are they, and then you can make an informed decision. And it's the same thing with the cannabis, to not just assume that crop to crop, batch to batch, that you're always getting the same thing, but each time along the way, during the agricultural phase and the processing and at the final product phase, to at the very least do chemical fingerprinting to see, do we at least have the same spectrum of bioactive compounds?
Exactly. Well, I've also read a little bit that you have been doing some research on cognitive function enhancements.
Yes.
Could you elaborate a little bit about it? Could you tell the audience what that all about, please?
Sure. So there are lots of studies, all the way from the shocking that I think it was at Harvard, somewhere in Massachusetts, a study was done, that showed that smoking marijuana actually enhanced cognitive function in adults, and also specifically what are called executive cognitive functions, decision-making functions.
Mm-hmm.
And that's surprising to people often, but again, remember, we've got to take into consideration the intent of the person using the product, because one, just saying marijuana or cannabis is really not a specific agent, like I said, like with, Viagra, or, with Lipitor or something like that. It's a family of compounds. It's like saying taking an anti-inflammatory, or it's like, there's a lot of diversity in what is in those final products. But even within that space, once you even narrow it down to a specific product and say it has a 1-to-5 ratio of CBD to THC. Fine, okay, and so maybe everybody's using the same one, but what's their dosage? And so if they're smoking, are they smoking to fall asleep? Are they smoking to get high, or are they smoking just to relax? And so I think that's why sometimes it can be confusing for people that there can be a beneficial effect on cognitive function. So one of the biggest challenges, and two biggest challenges in cognitive function, one, again, as I mentioned, is disinflammation or inflammatory dysfunction and the effect that has on the brain. And also that can originate even in the gut. And so if you have gut dysbiosis or gut disinflammation, the gut biome is in constant communication with the brain. And when there's an inflammatory process going on in the gut, it actually can trigger inflammatory response in the brain, not necessarily at the cellular level, but the molecular signal that's arriving to the brain is then telling the brain to change its neurochemistry to that of, if you will, a depressed person. It's kind of a very general way to say it. And so often people that have something going on in the gut, actually we recognize it in their behavior, in their affect. So depression and anxiety and mood disorders actually decrease cognitive function. They decrease executive ability. So sometimes it could just be about a tune-up of the digestive tract and the microbiome, and also the endocannabinoid system to break that disinflammation signal that's running back and forth. The optimal dosage is likely to vary from person to person. It depends on what's being produced in their gut. Maybe their serotonin production in the gut is depressed because they've got some, crazy bacteria down there that they picked up, from some fast food joint or something, or wherever it is that, humans are a dynamic, organism. It's like I was saying, there's a lot of biodiversity, but also there's individual diversity, meaning if I travel to Thailand and I stay there for 3 months, and then I, go to Argentina and I stay there for 6 months, my body is changing. And the job of the endocannabinoid system is to help modulate that change, what we call homeostasis and harm reduction, so that this organism, it's kind of like a smart device, like a smartphone. I want my smartphone to work just as well in Thailand and Argentina as it does here in Seattle. And so the endocannabinoid system is like that operating system. It's like that greater system that makes sure that all the functions of my smart device work in all of these different environments. And so my cognitive function might seem like it should have no greater challenge, in Bangkok than in Seattle than in Buenos Aires. But there are other things going on. And there are other things going on in my digestive tract. I'm going to be eating different food and I'm going to be shifting my microbiome because it's a different environment, and it's going to start to harmonize itself with a Thai microbiome, if you will, or, a Buenos Aires microbiome. And that's going to change the signaling that's going back and forth between the GI tract and the brain. So all of that, I know I make it sound very complicated. I apologize, but all of that is occurring.
I've been learning a lot. I wish we could continue this conversation like forever. Like I could pick your brain all day, but.
Well, you should take me out to dinner tonight. There's some great restaurants in Distrito Federal that I know of. Last time I gave a lecture there was 2 years ago, and I wish I was there. Me lastima mucho que no estoy allá con ustedes en Distrito Federal, but COVID and Zoom, and we're all in our little bubbles.
Sí, yo estoy desde Guadalajara.
¿Ah, sí? Ah,
sí.
Me encanta Guadalajara. Muy
Bien,
rica
bien.
la comida por allá también.
Bueno, doctora Aliffe, Karyemaitre, right? Did I butcher it?
Oh yeah, but butchering is the first step towards a great steak.
Okay. Sadly,
you've been grilling me for half an hour, so you should butcher me before you grill me anyway. That makes sense.
Okay, well, thanks a lot for this. I really love to pick your brain, spend some time with you. Sadly, we're done. We are done with time. I need to jump to the next interview.
Mm-hmm.
But it was a pleasure meeting you, sir, and I'm looking forward to working with you eventually someday soon.
Igualmente, el placer es mío y me gustaría mucho platicar más y trabajar con ustedes allá en México, mi México lindo y querido.
Aquí está tu casa.
Bueno, gracias.
Nos vemos.
Nos vemos. Hasta pronto.